woman or girl. The genetic material also revealed her ancestry. When researchers compared her DNA to ancient populations, they found that she had genetic affinities to a group of ancient North Eurasian individuals who lived around the same time period. But this population had been documented only further east, in Siberia. The woman who wore this pendant had come from approximately 200 kilometers away. She had traveled, or migrated to Denisova Cave, and during her time there, she wore this small piece of jewelry. The DNA provided additional information. When compared to modern populations, her genetic material aligned most closely with Native Americans. This suggested that her population lineage was ancestral to the peoples who would eventually migrate to the Americas thousands of years later. She was part of a genetic legacy that would stretch across continents and millennia. She was connected through the invisible thread of DNA to millions of people who would exist in a future she could never have imagined. The mitochondrial DNA from the pendant also revealed information about the deer tooth itself. Scientists could identify the species Wapiti, a large species of deer still found in Siberia and North America. They could estimate the age of the pendant based on the accumulation of mutations in the mitochondrial DNA. The pendant dated to approximately 19,000 to 25,000 years ago. It was genuinely Paleolithic. It was genuinely ancient. But perhaps the most profound aspect of this discovery was what it revealed about human behavior and identity. This woman had taken the time to obtain a tooth, to craft it, to drill a hole through it, to thread it on cord, to wear it against her body. Why? Was it decoration? Was it a talisman? Was it a connection to the animal she had hunted? Was it a gift from someone she loved? Was it a symbol of status or achievement? The artifact suggested a mind that valued beauty, that valued self-adornment, that invested time and skill in creating something that served no practical purpose beyond aesthetics or spiritual meaning. This was not a tool made for survival. This was jewelry. This was art. This was personal expression. And because she wore this pendant, because her skin cells and sweat and blood had contacted the porous surface of the tooth over time, her identity was preserved. Her genetic essence was recorded. 20,000 years after she walked the earth, science could recover her DNA and know something essential about who she was, where she came from, what genetic populations she belonged to. The pendant became a locket of her DNA, an unintentional time capsule, a message sent across 20 millennia. The research team was careful to note the limitations of their findings. The DNA proved that a woman had handled the pendant. It was likely that she had worn it, but there was a possibility that someone else had made it, and a different woman had worn it at a later time. The DNA could not definitively answer the question of whether she was the maker, the original wearer, or a later user. The genetic information existed in extraordinary abundance, almost as if the pendant itself had become a human artifact in its composition. But this abundance raised the possibility of contamination. Perhaps multiple individuals had handled and worn the pendant over centuries or millennia. The DNA recovered might be a mixture of multiple people from different time periods. But even accounting for these uncertainties, the discovery was revolutionary. For the first time, science had directly connected an artifact to a specific individual based on genetic evidence. The cultural record and the genetic record had been linked. An object in the archaeological record had been associated with a specific human being. An object that was created by human hands, worn by a human body, shaped by human decision-making, could now be understood not as an abstract artifact, but as something that had belonged to a real person. A person with a genetic identity. A person with ancestry. A person who had made choices about what to wear, what to value, what to keep close to her body. The implications for archaeology were staggering. Every artifact made from bone or tooth that had been touched by human hands now became a potential source of ancient DNA. Every tool, every ornament, every piece of jewelry, every decorated object, all of them might contain genetic material from the people who made or used them. The archaeological record had always been mute about individual human identity. Artifacts told us that humans had lived in a location, that they had created tools, that they had valued certain materials, but they told us nothing about the specific individuals behind those artifacts. Now, DNA extracted from artifacts could provide individual identity, could reveal whether an object had been made by a man or a woman, could trace genetic ancestry, could identify the specific populations involved, could create a bridge between the material culture that archaeologists study and the genetic history that geneticists reconstruct. The technique developed for this pendant was not unique or limited to this single discovery. The researchers showed that the method worked on other artifacts as well. It worked on bone samples from multiple caves. It worked on items that had been freshly excavated with proper contamination control. It worked less effectively on items that had been extensively handled by modern archaeologists or buried in certain soil types. But the potential was clear. Thousands of Paleolithic artifacts housed in museums around the world could potentially yield human DNA. Artifacts that had been sitting in collections for decades or centuries, unstudied and unexamined from a genetic perspective, might contain extraordinary information about the people who created or used them. The discovery raised fascinating questions about the nature of human identity across deep time. This woman lived 20,000 years ago. She belonged to populations that were ancestral to Native Americans. She lived during the Ice Age. She was part of the complex tapestry of human populations that occupied Eurasia during the Upper Paleolithic. She made or wore jewelry. She migrated or traveled across Siberia. She was an individual with a life story, a history, a genetic inheritance that would persist for 20 millennia. Now, scientists could begin to imagine reconstructing the lives of individuals from the deep past: a woman who wore a pendant, a man who hunted with a tool, a child who played with a carved figurine. All of these individuals, if they had left their biological traces on objects they handled, could potentially be identified. Their genetic identities could be established. Their ancestry could be traced. The abstract past could be populated with real individuals. The archaeological record could be humanized through genetic science. Yet there was another mystery lurking beneath this one, a darker, more troubling revelation. As scientists mapped the genetic transitions across different periods of Egyptian history, they noticed something alarming. Between the Old Kingdom and the Middle Kingdom, roughly around 2180 BCE, the genetic portrait of Egypt shifted dramatically. A new wave of invaders, people from the East, had swept into Egypt during a period of political collapse and chaos. They were the Hyksos, Asiatic peoples who dominated northern Egypt for several centuries. The genetic evidence showed that they had left a mark, a permanent imprint on the Egyptian population. Even as Egyptian kingdoms reasserted themselves, even as dynasties rose and fell, the genetic contribution of the Hyksos remained. It was written into the bodies of millions. It was a story of invasion, of foreign domination, of cultural resistance and eventual integration. And it had been hidden in the DNA for 3,500 years. But perhaps the most stunning discovery came when scientists looked at the DNA of the most famous pharaoh of all, Ramesses II, the great builder, the man who ruled for 66 years, who fathered over one. Researchers found something extraordinary. Ramesses carried genetic markers that were exceptionally rare in ancient Egypt, markers that were more common in populations from the Levant and Mesopotamia. But more remarkably, some of his genetic variants were associated with longevity, with disease resistance, with the biological capacity to father children well into old age. Science could finally explain how Ramesses had lived into his 90s, an extraordinary lifespan for the ancient world, and how he had continued to produce offspring for decades. His legendary fertility was not merely a matter of cultural polygamy, it was rooted in his actual genetic inheritance. His body, like his monuments, was built to last. The question of where ancient Egyptians truly came from became even more complex when researchers began examining the mitochondrial DNA of mummies. Mitochondria are the powerhouses of the cell, and they carry their own small strand of DNA that is passed down exclusively through mothers. By tracing this maternal lineage back through thousands of years, scientists could map the roots of female migration into Egypt, the paths that women and families had taken as they moved across continents. What emerged was a web of connections. Some maternal lineages traced back to Africa, others to Anatolia, still others to the Mediterranean islands. It was as if Egypt was a meeting point for thousands of different family journeys, each one carrying its own story, its own destination, its own role in building civilization. But here's where the story takes a turn toward the future, because understanding where we came from is not merely an academic exercise. It is the foundation for understanding who we are. And who we, countless conflicts and genocides, is revealed as fundamentally false. Every human population is a mixture. Every person alive today carries DNA from dozens of ancient populations, from migrations spanning tens of thousands of years. We are all hybrids. We are all, in a very real sense, the descendants of Egypt. Because Egypt represents something essential about human nature, our capacity to move, to mix, to create something new from diversity. Yet the genetic research also forced science to confront uncomfortable truths about the ancient world. The DNA evidence showed that Egypt's greatest periods of achievement, Greece, the earliest inhabitants of Mesopotamia, everywhere they looked, they found the same pattern. The great civilizations of antiquity were not the products of isolated peoples perfecting themselves in isolation. They were the products of interaction, trade, migration, and cultural exchange. The very thing that ancient historians and archaeologists had suspected from material evidence, that the Mediterranean and Near East had always been economically and culturally connected, was now confirmed in the DNA itself. Humanity's greatest achievements had emerged not from purity, but from mixture. This genetic revolution is continuing even now. Scientists have recently begun extracting DNA not just from mummies, but from bones embedded in ancient pyramid walls, from the mortar and clay used in, and perhaps most tantalizing, scientists are now exploring whether the genetic knowledge of ancient civilizations themselves might have been encoded in texts. Were the ancient Egyptians aware of their own genetic? In 1978, scientists were granted something that had been denied to researchers for centuries: permission. Permission to touch one of history's most sacred and controversial objects. Permission to take samples. Permission to study. Permission to apply the tools of modern molecular biology to an artifact that had spent nearly 700 years wrapped in mystery, legend, and faith. What they discovered in those samples would remain locked away in scientific archives for decades, analyzed but not fully understood, containing secrets that only the most advanced genetic technology could eventually reveal. That time has come. And what the DNA within those ancient threads has finally told us challenges everything we thought we understood about one of Christianity's most precious and contested relics. The Shroud of Turin is not simply a cloth, it is a mirror. It is the point where history collides with mystery, where science meets faith, where evidence confronts belief. For centuries, millions have looked upon it and seen a face, the imprint of a crucified man. And they have asked the same question over and over across generations and continents. Is this real? Is this the actual burial cloth of Jesus Christ? Is this the only remaining physical evidence of the central figure of Christianity? Or is it an elaborate medieval forgery, a work of human artistry and deception created to inspire faith and extract a devotion from believers? The shroud first appeared in the historical record in 1355, in the possession of a French knight named Geoffroy de Charny in the town of Lirey. Before that, its trail disappears into legend. According to tradition, it traveled through the ancient world, through Edessa in Turkey, through Constantinople, through Athens, through the medieval kingdoms of Europe. Each story grows more elaborate, more distant from historical documentation, more dependent on faith than fact. The shroud became a relic, a holy object, something that believers traveled thousands of kilometers to see, to touch, to stand before in reverence. Popes declared it authentic. Pilgrims wept before it. Artists painted its image. Theology was built upon the assumption of its truth. But in the modern era, science began to ask uncomfortable questions. If this cloth had truly wrapped the body of Jesus Christ nearly 2,000 years ago, it should tell a story. It should contain evidence. It should preserve traces of that ancient contact, protected by the very fibers that held the image. In 1978, a team of scientists conducted the Shroud of Turin research project, known as STURP. They examined every millimeter. They took photographs. They collected samples. They applied spectroscopy and chemistry and microscopy. And they found something remarkable. They found that every explanation seemed insufficient. The image itself defied conventional art techniques. No pigment could be detected, no paint, no dye. The image appeared to be created by something that oxidized the fibers themselves, browning them from within, as if the body had left its trace imprinted directly into the cloth at the molecular level. But the image was only the beginning. Locked within the fibers of that linen cloth were thousands of stories waiting to be told. Stories not of faith, but of chemistry. Not of theology, but of biology. Stories written in the universal language of DNA, the four-letter code that connects every living organism on Earth. For decades, those samples collected in 1978 remained in archives, studied but not fully unlocked. The technology simply was not advanced enough. The tools did not exist. But science does not stand still. Knowledge advances, capacity expands, and what seemed impossible 20 years ago becomes routine today. In 2026, a team of Italian researchers finally brought the full power of modern molecular genetics to bear on those samples. Gianni Barcaccia of the University of Padua and Alessandro Achilli of the University of Pavia led the investigation. They employed a technique called metagenomics, a sophisticated approach that allowed them to sequence every genetic fragment present in the dust and fibers, regardless of origin. Plant DNA, animal DNA, microbial DNA, human DNA, all of it, everything. They could not simply look for one specific source. They had to analyze the complete genetic tapestry, the accumulated biological signature of centuries of contact, handling, veneration, and storage. What they found was overwhelming. Not one genetic signal, but hundreds. Not a single source, but a chorus of voices speaking from the distant past. The data was so complex, so layered with contamination from so many sources that it told a story not of a single event, but of a journey. A geographical journey. A historical journey. A journey that the cloth itself had preserved in the code of life.